Hi everyone. So my name is Goncalo Regalo. I'm first of all, I want to thank the scientists for giving me the opportunity to be here, and I'm really excited to be sharing with you all a few thoughts on some considerations on how to design scalable cellular therapies from the beginning on to assure a successful development and translation into clinically ready final products, in a way making that transition from early research to reality in some of these advanced therapies, right? And the reason behind this is that if you look at the current landscape, right, there's a lot of potential in gene therapy and cell therapy. There's a lot of clinical trials, a lot of preclinical work with very innovative approaches to tackle, really nasty diseases that until now didn't really have a proper uh, um, approach and an innovative approach to tackle, right? However, if you look at that funnel, there's a lot of preclinical work, a lot of clinical trials, but very few actual approved cell and gene therapy. So there's a gap clearly that prevents these therapies from reaching patients, right? So this gap is, there's a multitude of reasons. But there's clearly a few factors, a few common factors that make this transition particularly complicated in advanced therapies in cell and gene therapy. Clearly, high costs of these therapies have been a factor, and the inability to take full advantage of the economies of scale when you look at small patient populations or an autologous setting, right? But there's clearly as well, particularly looking at cell therapy, the issues in terms of process control. It's a first in a way for the to use living modified cells, expanded cells, right? To manipulated cells as therapies, right? And to do so in a very consistent way, and manufacture them in a very controlled way while keeping the critical quality attributes of the therapies under control. It is a challenge, right? And all the deviations, the processing consistencies, the lack of scalability is proving to be a massive issue in the adoption and scalability of these therapies, right? So one of the challenges, and speaking to you all, coming out of especially in academia, working on the early stages of many of these future applications, right? And I think one thing is unique about cell therapy is that early choices in terms of the clinic, in terms of the raw materials, medias, the process design and architecture really do influence clinical outcomes, and are very tightly connected and with actual commercial outcomes, and the successful scalability and adoptability of these therapies, right? And 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 this is, in this particular case, it's essentially linked to the biology, and the need to reach particular biological outcomes, things that we do in research all the time, in a small setting, in an academic center. But in this case, it will have to be then translated into an actual pharmaceutical product, right? So questions of the starting material, variations in terms of the media, the cytokines, the process, and the way this affects the phenotype, right? And then the need to do that and resorting to high quality reagents and equipment to have that line of sight into the commercialability and scalability, and all the regulations that are underlining the approvals of these therapies. This is where this becomes particularly tricky. So this talk today is going to be a lot about how do we balance. It's a balance game in the end between the biology one wants to achieve, and then the compliance and the quality of some of the choices we have at our and you have at your disposal as your as future cell therapy developers or cell and gene therapy developers. It's very important, and this balance, in a way, it's very much behind the thought process that is expected for therapy developers in the cell and gene therapy space by regulators, which these products are advanced therapy medicinal products, ATMPs, and the process behind the decision making of in terms of these critical elements, it's a risk based, and to have a strong approach based on risk assessing these decisions from multiple ends is the way that is expected for developers to move forward, right? um, and this is when we look then at the biological challenges that are quite unique to these cell manufacturing processes, and try then to embed quality in the manufacturing setting, in the architecture, right? From the donor heterogeneity to media sensitivity to the variable expansion and variation in the quality attributes, in the phenotype, in even sometimes in the potency of these drugs, which are drugs in the end. These cells will be a pharmaceutical product, but again, as well, manual handling. It's in the end, a PhD will manage to do a CAR T cell relatively easily following a protocol. But all these very to do this at scale, we need to have this process is much more streamlined, reducing manual handling to make sure that this is then scalable, right? And this is then linked then with standardizing starting materials, using defined media and raw materials, a lot of in-process control, and it's very clear that this control has to be embedded in the manufacturing process, right? And then the big question of how do we automate? And automation and closed system is essential to cell therapy processes at scale. And this, arguably, there's different approaches to do cell therapy, so allogeneic, and for instance, but clearly autologous cell therapy, this is where it becomes particularly, it is a great example on these challenges, right? Because we're talking here about an intrinsic biological heterogeneity into these processes is embedded in that starting material that is patient derived, right? So prior treatment history, that biological heterogeneity from person to person, the collection outcome variabilities, all those factors will impact ultimately how the cells behave, the cell counts, the viability, the phenotype, the process parameters that one needs to control. And all of that is in essence, those critical quality attributes is the drug itself, is the potency, is the quality of the final product. So people will develop, there's different approaches to standardize this, by standardizing collection procedures, develop adaptive process architectures, explore process normalization opportunities. But clearly, if we can reduce if the starting material is very diverse and variable, if we can embed and if we can mitigate and offset some of that variability by resorting to define reagents and to automate the process, definitely that's the way to go. And when we look at that in terms of process architecture, and this is a very general cell therapy process, starting with a collection step, an enrichment step. If it's a T cell, these T cells might be activated, genetically modified. If it's a CAR T or a TCR-T type of process, expanded to a clinical dose, harvested, formulated, often cryopreserved, and then all the way to the bench side. So when we look at all these processes, we have to think about how do we keep this process under control from the start? How can we, maximize the outcomes? But by working with the different process steps, let's say enrich using one can start. One can actually expand T cells, activate them out of a mix of PBMCs, for instance, but that will definitely add different elements there that are much harder to control, particularly in an autologous setting. So enriching that with magnetic beads, starting with a more defined cellular population is, for instance, a strategy. Cell expansion, taking the cells up to a certain level, harvesting it in formulation, fill and finish is extremely important. But across the board, what we do believe is that we have to start thinking about reducing the variability of these raw materials and looking at automation. How can we reduce manual handling? How can we automate and close the processes as we move forward, right? So in starting with with the raw materials, clearly one of the most important materials in cell therapy manufacturing is cell culture media, right? And when we take a bit, as I said before, a risk-based approach to this, and we look at elements that are typically used to supplement or that are intrinsic parts of cell culture media, particularly when we start in academia developing these processes, for instance, animal serum, fetal bovine serum, is commonly used, but this is obviously something that will support biology really well, but then will contain and will entail risks in terms of the viral content, the product variability, that in principle will make therapies that are based on animal serum supplemented media is extremely challenging. So people have to start looking at different alternatives like human serum. But when you look at human serum, that again also has risks, and there's if you look at the regulatory landscape, there's a lot of conversations, particularly in Europe at the moment, on human derived components, and how do we trace back to the donor, how do we assure that you are working with suppliers that where that human serum is properly sourced, right? So looking then at, there's other things, the quality of growth factors, elements that are commonly added to media like phenol red that can cause actually allergic reactions, or antibiotics for the same reason, right? So all of these entail risks, and these are all tools that are commonly used in the lab and in early step development. So one thing that very clearly, very early in the process, and for the ones of you out there looking at making or developing a cellular therapy, one very early step that what needs to be taken into consideration is really try to de-risk those raw materials, resorting to either serum-free, animal-free, xenon-free media formulations, or ideally a chemically defined cell culture media where all elements of that media are defined, which assures that a reproducibility in terms of the raw materials of that process, and that biology that one needs to achieve at a certain process step, right? This is extremely important, because in the end, media can be a manufacturing strategy, like not only just growing the cells, but the way that media impacts the cell expansion, the phenotype, how it impacts reproducibility. At the same time, using those chemically defined formulas, reduce lot-to-lot variability, improve the outlook on scalability, enable regulatory consistency across the board. This is definitely something that will, pay off in the long run as these therapies are being developed and through the clinical approval process. However, of course, it's up for each one of you and each developer to make that assessment. Do I reach the biology with the media I need? Do I need to supplement it further? And then it's a conversation, and how do we, and a justification from a risk perspective. Does that media enable me to reach the biology I need at scale, or do I need to make a compromise? And all is possible, but it has to be very well justified for regulators in the end, right? So what is clear is that whatever choice you make of raw materials in an early stage will impact your development further down the line. If you lock your precious biology, your critical quality attributes of your cell therapy process, drug product, uh, early with poor quality research grade materials, serum, fetal bovine serum containing medias. This will greatly impact down the line the scalability strategy. Might force you to do eventually expensive comparability assays to assess that when you try to improve the quality midway of some of these reagents. So some of that is possible and desirable and will happen, but in terms of the critical materials, that has to should be defined as early as possible. Another element I wanted to call the attention is, and it's quite unique to cell therapy when you look at in terms of the in terms of the biopharmaceutical manufacturing, is that cells cannot be end sterilized. Right? So you can't irradiate cells, you can't filter cells, right? So the expectation from regulators is that aseptic manufacturing and is embedded into the process. And if you're working in an open environment, manually, for instance, pipetting things in and out to your culture flasks, right? That in the future will require you to have the most stringent clean room setting available, which is actually a grade A laminar flow on a B clean room background. Complex gowning, extremely expensive, a pain, absolute horrible to work with in now, if you resort to automated systems and these cells are processed in an aseptic environment, single-use systems, this in disposable systems, and it's considered a fully closed system. Now, the limits of operation in terms of this clean room drops dramatically, which means it sounds cheap and easy to manufacture cells under the laminar flow, pipetting things in and out at first. But when you look ahead and you want to start making these cells at scale up to a certain degree, very quickly that transition will need to be made because it's extremely complicated to handle this in a scale way. And at the same time, there are quite a lot of interesting automation pieces that help people fully close and automate these systems, these process steps to achieve full control and full aseptic closed manufacturing, right? And this is one of the elements I think, definitely you as developers are spoiled for choice. There's quite a lot of automated systems out there. There are systems that even provide you a complete full box end to end with a single piece of equipment. These systems play a role, and they're great to get your foot on the door on some of these automation type of approaches. If you're not fully familiar with that, it's an easy entry. But however, when we look at how do we rationally scale processes in the future, these single boxes become a little bit less appealing, and we do strongly believe that a modular workflow design is desirable. So pretty much going step by step, cell selection, cell expansion, fill and finish, right? And understand from the automation landscape, from the providers of automation, what is the best piece of equipment that will enable my teams and me to reach the best outcome and hit my critical quality attributes at a particular process step, right? And indeed, those in-process controls and the type of data that these systems generate are going to be invaluable tools to keep the process under control, right? This is, for instance, one example of this, for instance, is T cell expansion. And we know from our experience in our labs as well that, T cell expansion in chemically defined conditions it's absolutely possible and desirable and with high densities. But there are some considerations one needs to take when building that seed train from a very small cell number to large, to the modification one needs to make on these cells, the activation, the transformation, the modification, and then reaching expanding these cells to high doses. And right now, this is often done from culture plates to gas permeable systems, then all the way to bioreactors. And there are several options out there. So one has to have a very critical look on how do we build this to make sure we don't grow these cells in environments that are ultimately a little bit challenging, and to keep the cells and again the process as under control as possible. An example of that is the way gases are distributed in these different platforms. I know many of you out there will be working with cell culture plates and flasks throughout most of your early development and academic career, so to say. But from a biological standpoint, with a top-down gas distribution, it's actually quite limiting, particularly for T cells, from a biological standpoint. And this becomes particularly challenging, again, if you remove that serum, extremely rich, extremely diverse source of nourishment and growth factors, right? If you remove that those elements become a little bit tricky. So some of these process changes are very much possible and desirable, but need to be taken that need to be made in a rational way, right? And then if you scale, let's say, to a stir tank bioreactor, is the biology that I achieve in my culture plates actually translatable to such a different system where the cells are stirred, might have shear stress, right? associated, or even a rocking motion, which again has a different way of aerating cells and T cells. So if you do your small manipulation, your small scale manipulations like transformation activation, and then you inoculate your rocking motion bioreactor in chemically defined conditions, how does this, how this will this work and impact that biology? Right. So it's really important that these choices are made in a rational way. There's of course the point of adoption. Where are we going to adopt? When is reasonable to start adopting some of these automated systems, and a lot of them are very expensive, obviously. So there's that considerations need to be taken at that level. But I think from a just simply on a process design, there definitely are considerations one needs to take, particularly on that, flask, plates and flask biology, like very labor intensive. It's really locking in a biology that in the end is going to be really hard to scale. In our hands, we are big fans of hollow fiber bioreactors. We do think from a T cell perspective, they do lead to semi-physiological ways of these cells to grow in the fibers that are leading to high densities and optimal phenotypes in what T cell is concerned. But there are many options and different strategies. Let's say if you want to do stir tank bioreactors in the later stage, perhaps don't start with a flask in a small scale, but start with shaker flasks, or with a small scale bioreactor. Now there's plenty of small scale stir tank bioreactors. There's right now extremely different options to do that. So take in consideration, think ahead, and even if you do small scale and not really fully automated, start thinking, how do I then going to translate that into my platform of choice later down the line? In our case, and we regularly use expansion with our chemically defined T cell media in the quantum hollow fiber bioreactor. And it's a great example where in about 7 to 8 days, you start with a very small cell number, and you can achieve up to 6 billion cells, and with optimal biology, and you see the control, the nice drop in glucose and rise of lactate as the cell progresses in a very linear fashion. So we do think this particular combination is particularly useful. But again, this is something you will have to assess on a case by case on your systems, on the bio. Try to see what's best out there for the biology you want to achieve, right? Whatever your choices in whatever your biology, just really design for scale early. Right. So select defined material, media material, and qualified raw materials that you can procure and that will keep your process under control, whatever the stage you're in. Right. Develop scalable cultural strategies. Look at the process architecture. How do you reach that scale? Even if you're not there yet, think about it and plan ahead and align the workflows also with future GMP requirements. Right. And identify high variability in labor intensive steps, and design processes compatible with this scale out, and as much as possible, as early as possible, particularly in relevant process steps, like cell expansion, or as in magnetic bead enrichment selection. Implement these closed system and automated platforms whenever relevant, as early as possible as well. And I think this is really the equation for successful translation. And how do we couple then those early stage decisions through process development all the way to downstream success, right? As far as we are concerned, we do think, and I know the relationship between vendors and developers is quite different in academia. I think in early stage developments, reach out to your provider, in our case, and to Fujifilm Bioscience, and or whatever this provider you have for your ideal process step, because in principle partnership matters, right? I think only vendors and developers working together can we actually work on developing these more future proof reproducible processes, right? And then I know there's the element of cost. A lot of this is expensive, but I think we're reaching a moment in the industry and in the maturity of cell and gene therapy, which these conversations are all possible. So just reach out to your to your provider, and find the right partner to support you at each manufacturing step, and to plan ahead, and have line of sight of the future, right? In my case, this is me, Goncalo. This is my email. Would be really happy to reach out if you have any questions. We'd be happy to tell you a little bit more about what our thoughts are in Fujifilm Biosciences and how we make this a reality. And yeah, that's it for me today. Thank you very much. Okay, so we have a few questions coming in. So yes, one question I see here is like, how early should we realistically start thinking about GMP compatible raw materials, and what are the trade-offs if we wait too long? And absolutely, people can use research grade reagents up to a certain point. There are developers that offer pre-GMP formats, for instance, to that will lead, where you can pretty much work with a pre-GMP formulation that is then more or less translatable then into a GMP setting. In our case, we like, in the Fujifilm Biosciences, again, philosophy, we love to pretty much work with you, and see how can we make sure that not only the products are the ones you need, but they're at the price point you can afford them, to make sure we support with the right products at the right point. The trade-offs, if people wait too long, is clearly that at some point, um, either a regulator or, at the latest at the IND, people will start asking difficult questions, and at that point might be too late. It will force you then to either go back to the basics or reinvent the manufacturing setting, or enter really complicated conversations between your provider and your regulator, to try to assure that they are satisfied with the responses and the strategy. So, something to take into consideration. Um, Let me see. Okay, another question: where do teams typically run into challenges when translating early process development into manufacturing, and what could have been done differently up front? That's a great question. I think it's really understand what makes a cell unique at a given point, right? Um, there's a lot of emphasis on in from a regulatory perspective into in process controls, right, where one understands the modification of the cells, and we don't wait until the at the end of manufacturing to assess if the cell is sterile, if the end product is correct, is exactly how you want to have it. Usually those are outcomes in a few weeks time. So the idea is that we embedding that quality and that process control, and that in process monitoring, is one way of keeping that at bay, right? But clear, of course, as I said, these challenges will be adopting chemically defined media, for instance, is a different mindset almost, right? I don't, we do, in academia or in early process development, like we emphasize on performance, maximum cell yield, right? A lot of times, adopting these high quality reagents, this serum free, these chemically defined reagents will imply a certain nuanced approach to what the outcome is, and that is going to be, it's something that can be challenging. But I think this is again where you have to work with your with your vendor, with your service provider, to maximize the outcomes, right? And let me see. At what point does it make sense to start designing processes with automation or closed systems in mind? Um, yeah, absolutely. I think, as I said, that there are processes that are intrinsically, um, that can be done early on in an automated setting. And I did mention magnetic bead separation, for instance. It can be done manually, but very, definitely there's an element of automation that's pretty great automation out there for this particular process step that will enable people to adopt that. And being that such a critical process step, this is one that makes sense to start implementing early on. Bioreactors, of course, will be something that will make sense to implement, right? I did mention the single box solution. These are a few out there as well that are great. As in, uh, the rational approach to this is in terms of the scalability, is that basically these systems do have the cell isolation enrichment module, a cell expansion module, and fill and finish module. And technically, you use the cell separation module only on day one, and the harvesting on the last day of the manufacturing. Um, which means while the cell expansion is the longest part of the process, and this will be where your cells will stay the longest, which means in this case you will actually be locking in your whole system, including the cell isolation and cell separation modules that will stay idle while the cells will be on the expansion module. So in terms of process rationality at scale, this becomes a little bit challenging down the line. Thank you, everyone. Thank you for the questions. Thank you for the engagement. Thank you for your time to listen. As I said, this is a process. Cell and gene therapy, cell therapy has unique potential from a clinical standpoint. We do strongly believe at Fujifilm Biosciences we need to act collaboratively, not only us vendors, but together with other vendors and other service providers, but also with you out there, at the front of the innovation, with you in your labs and companies, and just make sure we can make this a reality, right? I think the future is bright, but now it's really a moment where we need to rally together and try to address some of these challenges and do prepare for the future, because there's really a unique potential here that cannot be wasted. So thank you very much, and do reach out if you have any questions.